Assalamualaikum warahmatullahi wabarakatuh. Hello everyone, I'm Zahratush Shalehah from Biology Department, Faculty of Mathematics and Natural Science, Universitas Negeri Semarang. At this time, I will present a laboratory report on brain anatomy with the title Deep Meristem Tissue of Coleus sp. In this discussion, I will present it into 5 points, namely the introduction, objective, method, result and discussion, and last is the conclusion. Let's get started with the first point. This is an introduction. Shoot apical meristem, the shoot apex is the part directly above the youngest leaf primordium, which is meristematic. Apex shape should form an elongate direction, generally slightly convex and variable. Shoot meristem can be differentiated into promeristem and underlying meristematic regions, whose cells undergo certain stages of differentiation. Promeristem contain bikinary shoot together with their preceding cells and are still near the buds. The part that differentiates in the meristematic regions is the protoderm, and from this the plain epidermis system develops. The procambium, which will form the primary vascular network, and the ground meristem, which will form the plain ground tissue, such as parenchyma and cortex, and pitsclerenchyma and cortical collenchyma. Next, the second point, what is the purpose of implementing this practicum? The objective of this practicum is to observe the anatomical structure of shoot meristem on the object of Coleus sp and knowing the location with function of the apical meristem. Next. Third is method. What method is used? In this practicum, I will explain 2 types of methods that can be done. The first method is experiment, and the second method is image data analysis. The first method on the next slide. Okay. The first is the experimental method. Here is how the preparation of preparate and practicum lab work using the experiment method. First step is take the tip of the young Coleus plant stem about 1 cm long and color it with safranin. Second is make preparation of a longitudinal slice of Coleus plant stem with water observation medium. Third is observe under the microscope, from low to strong magnification, and less is picture and complete with description. Next, the second is the image data analysis method. Here is how the image search works and the practicum flow using the image data analysis method. First step is read a quiet but procedural practicum. You can read the manual that has been provided regarding the theoretical basis and procedures for making preparation. Second step is looking for pictures of observation object. You can looking for pictures of research preparation that have been carried out through practicum journal and other official websites. The third step is analyzing the part structure of observation object. You can analyzing the structure of the object part of the preparation. In this practicum, We were asked to observe the shoot meristem on the stem deep preparation of Colliers sp. And the last step is draw the pictures of result and write conclusion on the worksheet of laboratory report. You can draw the result of observation of image data of time on the practicum report worksheet with the format provided, and you can write, make the conclusion obtained from the practicum based on the introductory material on the practicum report worksheet. Next, continue is the result and discussion. Based on the result of practical observation, the result of the anatomical structure of meristem tissue on the object of observation of stem tip Colliers sp as follows. First is protoderm. Protoderm is the origin of skin tissue or epidermis. The precambium will differentiate into a transport network system. The precambium is the part that will form the primary vascular connective tissue, such as primary xylem and primary phloem, and the cambium. The function of the protoderm is to form the epidermis and precambium. Second is precambium. Precambium located in the middle with longer cells, and the precambium will form a network of xylem vessels, phloem, and vascular cambium. Third is ground meristem. The ground meristem is the tissue that will form the pit and cortex. The ground meristem is the part that will form the basic tissue or parenchyma of plants. Growth occurs because the activity of primary meristem tissue is called primary growth. Fourth is lateral meristem. The lateral meristem is a young tissue that is formed by initial cells located between parts of plant tools, namely between tissue, matter, tissue. Lateral meristem is the cambium cell which form the stem vessel, sometimes called the vascular cord cambium or vascular cambium. The cambium is a layer of meristematic cell or cells that continue to divide, which function in enlarging plant. With activity of the meristem, the plant will experience a large addition to the site. This lateral meristem activity will cause them and roots to increase in diameter. Fifth is axillary buds or lateral shoots. Axillary buds are located and develop from the axillary on the surface of the stem whose growth will expand laterally from the plant stem, forming branch or flower. And the last is apical buds. The apical buds are formed from the apical meristem, which function to produce new tissue on the stem, increase growth so that the plant increase in size. Next, and the last point is the conclusion. The purpose of the practicum is to observe the anatomical structure of shoot meristem on the object of Colliers sp. Colliers sp is a genus of the Lamiaceae family, and most are herbs, so they have stems that are soft and juicy. The result of the observation that have been made, it is that the stem deprivation of Colliers sp have anatomical structure in the form of basic shoot meristem, lateral meristem, apical buds, lateral shoots, protoderm, and procambium. Okay, I think that's enough That's all for me that I can convey. Sorry if there are mistakes in the delivery. Thank you. Wassalamualaikum warahmatullahi wabarakatuh.